Thanks for coming, and I'm sorry to those who attended the educational workshop yesterday because this will be a little bit familiar, just abridged, but maybe that means we can have a really fulsome discussion at the end of this panel. So today I'm going to be discussing how we've been using resting state functional connectivity to cluster people with major depression into subtypes, and how that might be used to inform who gets what treatment, be it TMS or other treatments like pharmacotherapies. So it should come as no surprise to this audience that depression is a very heterogeneous disorder. Many symptoms within the DSM-5 can be kind of the opposites of one another. Increased appetite versus decreased appetite, insomnia, hypersomnia, agitation, or psychomotor slowing. And as a result, there are numerous combinations of symptoms that can yield the same unipolar diagnosis. As a result, it should further come as no surprise that many people who are given first or second line treatments don't respond to the first intervention they receive. And in fact, about a third of people suffering from depression won't get better after 3 sequential treatments. How have we been tackling this heterogeneity? Well, in the past, many have took this top-down approach, looking at common self-reported symptoms, as you can see on the right, clustering people into subgroups like anxious or psychotic depression as specified by the DSM, and then looked within those subgroups to try and identify differences. And whether or not those biological differences may yield, information about what treatment one should receive. And unfortunately, that hasn't yet been meaningfully or individually predictive to date. So what I and others have tried to do is take a more bottom-up approach, integrating brain-based measurements during the subtyping scheme to see whether or not that might be more informative than just clinical self-report alone. Now, what we've been using is something called canonical correlations analysis, and in this case, we take resting state functional connectivity taken pre-treatment. We have clinical symptomatology. Typically, we use an assessor-rated scale, like the Hamilton Rating Scale for depression. And what canonical correlations analysis will do is essentially try to find linear combinations of these two modalities that maximize the correlation between these two latent scores. And that correlation we call the canonical correlation. We can use these coefficients that CCA will define with new data and then assess the canonical correlation with this new data, with a greater canonical coefficient yielding more variance explained in test data and a better fit. Now, previously, we've used this kind of approach in combination with hierarchical clustering to delineate both continuous dimensions explaining co-occurring symptoms and baseline functional connectivity, and hierarchical clustering to define subtypes. And this original paper from a few years ago, what we found were 2 dimensions, one explaining anhedonia and another explaining insomnia and anxiety, which clustered patients with depression into 4 subgroups. These subgroups were stable at a subsequent scan if they were still depressed, and they were predictive of antidepressant response to TMS, and in this case it was dorsomedial TMS. But there were some notable limitations of our paper, including the fact that we did not use regularization. Another limitation was that it was a multi-site secondary analysis, which may have limited our ability to generate stable or reproducible subtypes. We did not use regularization, which may have led to overfitting. And we did restrict our search of clinical symptoms to the 17-item Hamilton Rating Scale, which is somewhat limited in terms of how we can characterize certain symptoms, such as anhedonia, which is deficits in reward processing. And so the aims of our secondary or follow-up to that initial paper was to assess the performance and stability of regularized canonical correlations and hierarchical clustering in a large sample and further characterize these dimensions and subtypes using resting state functional connectivity, and then to see whether our updated model predicted antidepressant outcomes to TMS. And in this case, we were very fortunate. We had a very large, over 300 individuals with depression, single site sample of individuals with treatment-resistant depression who were all referred for TMS. To look at dimensions of symptoms, in this case, we did use the same 16 items of that Hamilton rating scale. We always omit the last one, insight, because most people, when they come referred for TMS, have insight into their disorder. But we also included 5 additional composite scores that encompass things like guilt, pessimism about the future, liking and wanting rewards, and irritability. So after training, which was stable and generalizable, we were able to discern 3 dimensions explaining co-occurring symptoms and baseline functional connectivity. And if you're curious to learn more about how we did that, please come talk to me. I just, for the sake of time, had to gloss over that. So our first dimension was related to increased low mood and somatic symptoms like decreased appetite and fatigue. This particular dimension was associated with basal ganglia and thalamic connectivity, as well as had top nodes in regions like the posterior cingulate and dorsolateral prefrontal cortex. The second dimension was related most specifically to anhedonia, with the top loadings you can see in sort of second to the left. And so rather surprisingly, it had top regions in higher order visual areas, such as the fusiform, as well as sort of a cingulo-opercular network and sort of the posterior regions related to that, such as TPJ, dorsal ACC, dorsolateral prefrontal cortex, and some more canonical reward processing regions like the orbitofrontal cortex. Now, the third dimension was most strongly associated with symptoms of insomnia, and it had top loadings predominantly in sensory motor cortex. Interestingly, a reviewer asked us to take a look at medication use in this sample and whether or not it covaried with any of these dimensions. It didn't with the first 2, but sleep aid use was associated with this dimension, which I found kind of interesting. And it will become more interesting in a second when I show the clustering results as well. Now, the features that were, the resting state fMRI features that were associated with each of these dimensions were relatively distinct, as you can see by this Venn diagram. And many of them were not atypical if we were to look at atypical resting state functional connectivity features relative to a large sample of healthy controls, indicating that features that predict inter-individual variability in symptoms need not be atypical. Now that we have, showed the continuous dimensions explaining co-occurring symptoms, we next sought to use those dimensions to define categorical subgroups and evaluate the stability and robustness of those subgroups. In this case, what we were able to find was basically a 4-cluster solution was stable. I want to see if I can show this in any way. So a 4-cluster solution as defined by the Kolinsky-Harabaz criterion was deemed to be the most robust, but only when we used those first 2 canonical variates, the first 2 dimensions. If we were to include the third, we would not find a local maxima like this, and it was not significant. At the time, as we submitted this paper, we weren't totally sure why, but after the reviewer asked us to take a look at medication use, it kind of helped explain to us in our minds maybe why the 3 dimensions did not successfully define subgroups, and the 2-dimension solution did. We also permuted this and found that this 4-cluster solution was significantly finding homogeneous clusters relative to what we would expect by chance. We also looked at basically taking subsamples of our data and using clustering again in the subsample, doing this 1,000 times, and then using adjusted mutual information to see whether or not cluster assignments were stable. So were the same people being grouped together in this 4-cluster solution. And indeed, the 4-cluster solution was not only beyond the elbow of this graph, which is what you're looking for with mutual information, but it was also significant relative to when we permuted the data. And this is just what it looks like as depicted, not just by a dendrogram, so you can see the 4 subgroups here, but also along our 2 dimensions. So what did these dimensions look like in terms of clinical differences? There were a number of clinical differences, and I'll just walk you through some of the trends. The first trend was that subtypes 1 and 2, depicted in navy and sort of a lighter blue, had higher mood and somatic symptoms predominantly. So increased self-reported low mood, as well as, here we go, GI symptoms and fatigue. Subtypes 2 and 3, in blue and sort of this mustard color, had increased predominantly anhedonia, so on these 2 composite scores that we created. So lack of interest and a lack of enjoyment. But they also had some deficits also on things like concentration and irritability. The 4th subgroup in red generally had the least severe symptoms across the board. Now, I won't, for the sake of time, show what was atypical across these 4 groups, but I'm happy to walk you through that if you do have questions. But I will show what we found with respect to response and remission rates to TMS. Now, in this case, we had 4 different kind of conditions. People treated with dorsolateral prefrontal cortex stimulation or dorsomedial prefrontal cortex stimulation, and with 2 different kinds of stimulation parameters, both excitatory, theoretically, 10 hertz stimulation and intermittent theta burst. And what we found, both using the categories, the subgroups, as well as continuously, was that anhedonia was related to improvement, such that if you had higher anhedonia or that you belonged to one of the subgroups that had higher anhedonia, you were less likely to respond to TMS. And it didn't matter whether or not you received dorsomedial or dorsolateral, 10 hertz or intermittent theta burst. It was really this anhedonia dimension that predicted outcomes to all of those possible interventions. So you can see subtypes 2 and 3 here having lower response and remission rates relative to subgroups 1 and 4. And this is also depicted continuously across the anhedonia dimension as a logistic regression with remission rates. Now, we were also very fortunate to have independent data of individuals treated with an SSRI. In this case, it was individuals across 6 sites treated open label with escitalopram for 8 weeks. And all we wanted to know was whether or not our clustering solution was replicable in this sample and if it also predicted outcomes. So what we did was we took this sample of about 130 individuals. We standardized the data with the means and standard deviations obtained using the training data set of 328 individuals, and we projected the data into the CCA space that we had previously published. Now, all we wanted to ask was, basically, are we able to replicate aspects of our clinical loadings? So did subtype dimension 1 also associate with mood or suicidality or whatever, or did subtype or dimension 2 also, was it associated with anhedonia? And then we also wanted to know whether or not cluster assignments predicted outcomes to SSRIs. And in this case, we were able to replicate aspects of our clinical loadings for both the first and second dimensions. And in this case, we didn't have the Hamilton rating scale for depression in the sample, but rather the 10-item MADRS. What we were able to find was that the first dimension, I didn't show this earlier, but suicidality was also a significant clinical loading on that first dimension, which we were able to replicate in this independent sample. And we were also able to replicate correlations with anhedonia. Interestingly, we were also able to find differences in remission rates to SSRIs, such that subtype 1 had the best remission rates, not only to TMS, but also to SSRIs. Interestingly, subtype 2 also fared very well to SSRIs in terms of their response rates. And if you looked at different patterns of symptom improvement, they actually saw some of the greatest improvements in their anhedonia. So to summarize here, subtype 1 does very well to both TMS and SSRIs. Subtype 2 does really well to SSRIs alone. Subtype 3 doesn't do great to either, and subtype 4 does fairly well to TMS. Giving us the potential testable hypothesis that if you're assigned to subtype 1, perhaps you should be receiving both SSRIs and TMS, which, according to some meta-analyses, have shown some promise in augmenting outcomes in TRD. So to summarize, and this is my last slide, is that given the right data set, these subtyping approaches are not only generalizable and stable, but they can yield us meaningful predictions in terms of outcomes to TMS and SSRIs. But it's important to note that this is only one potential subtyping solution, and not all data sets will be well-tuned to these kinds of approaches, especially in cases where you may have smaller sample sizes, idiosyncrasies with respect to inclusion or exclusion criteria, or weak associations between your clinical dimension and your imaging measures. So with that, I'm happy to talk more, and thanks for your attention. So there's one question. So the question is about how I would decide a specific target for a specific symptom cluster using the results. So how we're doing this is, like, this is all retrospective, but if we were to do this prospectively, we would use a very large training set to design this model, but we have these coefficients that we can use to get CCA scores for new data, and then based on the centroids of our clusters in our two-dimensional space, basically where they sit in that Cartesian space, based on Euclidean distance, we can basically assign a subtype. So whichever cluster centroid they're closest to is where we would decide that's their subtype assignment, and using the data that we've already previously collected, then maybe we can decide on what type of target or treatment to deliver for that new individual. I hope that answers the question. Thanks. Location? Location in the brain, so so basically using the results that we found, we actually didn't find differences by site. So in this case, you could probably flip a coin with DL and DMPFC using this model. The issue, right, is I know that there are, Sean's work and others have maybe suggested that there might be some subtype-specific symptom-specific differences in dorsolateral and dorsomedial, but this is only looking at baseline symptoms, not symptom-specific improvement. So you can use symptom-specific change and baseline functional connectivity to define clusters of symptom-specific improvement that might then yield more meaningful insights at who should get dorsomedial versus dorsolateral. So this is just one solution. There could be better or different solutions tailored to figuring out what TMS target specifically one person should be getting. But yeah, point well taken. Okay, thanks.